Hello, my name is Ann Lawrence. I'm the 7th grade science CTL, and this is the virtual unit preview for Unit 4. I want to start with a situation. So let's pretend that NASA wants you, in this case you're a genetic engineer, to colonize a planet. What kind of animal would you design to survive? What adaptations would it have? So let's talk about a potential planet. Let's say that NASA discovers a new planet and it's called Liroma. Let's describe this planet. This planet is dry and hot. Most of the planet is flat. Water is found in underground streams, but there is very little water on the surface of the planet. Most of the planet's surface is covered in sand, although there are patches of dry grasses. When plants get their roots down into the underground water, they grow into small trees with leaves at the top, but not along the trunk. Plants are not connected to the underground water, are small and dry, but are edible. Animals on this planet include insects, a species of birds that roost in high trees, and sand-colored lizard, and a small type of rat. So with this in mind, with this environment in mind, what kind of organism might you add to this planet to make it more biodiverse? How would we continue to colonize this planet? One idea that I had, and shout out to Andrea, the 8th grade CTL, for this image, is I could colonize or continue to colonize that planet with a adapted bunny, if you will. This bunny has especially long claws so that it can hold on to and run through the sand away from predators and keep more grip on the ground. It also is a relatively small and herbivorous animal, so the vegetation that exists or the little vegetation that does exist on this planet will be appropriate for this animal. And then the tail has a stinger on the end that will help it protect from the lizards and the rats and the birds that do exist on the planet currently. So in general, this unit, and we just exemplified this, is about figuring out what kind of adaptations or what traits are necessary in an organism to best suit the environment that they're in. Again, this is going to be a lot about natural selection and adaptations and evolution. Here's the agenda for this unit preview. I'm going to go over the priorities for CA number 3. I'm going to go through the order of objectives in the unit plan. I'm going to go over key misconceptions that students have during this unit. I'm going to explain the project in greater detail. I'm going to go through 3 of the labs that are in this unit, and then I'm going to, in general, talk about the end goals of Unit 3. That should say Unit 4. My apologies. Okay, so priorities for CA number 3. If you look in the top left-hand corner, here's like a real simple question. Students need to be able to know, or like to recall, where our genetic information is in the cell, and all the different names that could exist for DNA. It could be DNA, chromosomes, genes, all of this. Underneath that question, we have the differences between asexual and sexual reproduction, and how it connects with what offspring look like, whether they're genetically identical or different. So they need to have a solid understanding of where is our DNA, what's the difference between asexual and sexual. And then these top examples, both are pretty heavy text related to the adaptations of an organism. So students need to be able to, 1, comprehend more dense text in questions and apply what they learn in the text to answering what changed here, or why did they have this adaptation. And then down here, again, this is another example of you need to know where the genetic information is. And then over here, students need to be able to read a dichotomous key. All the questions on CA number 3 revolve around these topics. Where the DNA is, asexual versus sexual, in general adaptations and natural selection, and reading a dichotomous key. The constructed response looks something like this. In general, students need to be able to look at a situation describing natural selection and then make inferences and apply their knowledge. In particular, they need to be able to apply this knowledge to data tables. That's been a theme throughout all of our CAs. There's more data to analyze for students, so it's important that even though all of our specific data analyzation happened in Unit 1, it still needs to carry out throughout all of our units, because science doesn't mean much without data. So students need to be able to analyze it and come to conclusions based on that data. So that being said, let's take a look at Unit 4. Our statement of inquiry here is humans have the ability to change the natural world. So looking at different changes over time, natural selection, selective breeding, heredity, all of these things are included in Unit 4. This unit is actually a little bit shorter than the other ones, cells and human organ systems before this, and it will split its time across the winter break if you're on the same schedule that is in the scope and sequence. Just like always, there are two kinds of assessments. There's a regular paper pencil test that has been uploaded based on MYP criteria, as well as a project. And the project is very similar to what I started this video on. Basically, students are provided with a couple of different planet options, and then they design a organism and describe how that organism is best fit for their environment. The MYP criteria rubric is down at the bottom, included in the unit plan for your use. And the last thing with regards to the unit plan that I want to talk about is the order of objectives. We ended Unit 3, Human Organ Systems, with the reproductive system, and that usually gets students really prepared and ready to talk about asexual and sexual reproduction. We head into DNA, genes, and chromosomes, and so that it's easier to talk about the increase of particular genes or traits in natural selection, selective breeding. There's a quiz, just like always, in the middle of the unit. And then I have students do the strawberry DNA or extract strawberry DNA right before Christmas break or winter break. I've found that students are really engaged, and it's really hard to get students to pay attention at that time of year, and doing a lab really helps them solidify what DNA really looks like, how small it is, and it gives students an opportunity to get their hands dirty with some, more scientific tools. So that's basically it for semester 1, and then you do a CA review. And then after the winter break, then students come back and do one more main objective, reading dichotomous keys, which students usually pick up pretty easily. And then we head into the project and assessment. So again, this is a little bit of a shorter unit, but it is kind of difficult to get them to retain all the information from first semester over to second semester. So if you need to add a day in here for review, I highly encourage it. Let's talk about common misconceptions. When students learn about asexual versus sexual reproduction, a lot of them assume that asexual reproduction is cloning, and cloning really is a much more difficult and arduous process than asexual reproduction, so it's important that students make a distinction between asexual and cloning. While they both have the same concept as far as producing offspring from a single organism, the process for which they do that is different. Usually on this day I take an opportunity to introduce Dolly the sheep, but sometimes that can cause this misconception to happen, so you have to be careful about what topics you choose to read about. Students get DNA, genes, and chromosomes all mixed up as far as what is part of what and where is it. So students need lots of practice, and lots of pictures to reference. I have students draw the difference between DNA, genes, and chromosomes all the time, and put them in order, backways and frontways. A lot of students think natural selection can happen within one generation or overnight. It's really important that you make it clear to students that the slowest process would be natural selection, the next slowest process would be selective breeding, and then genetic engineering would be the most quick. But natural selection and selective breeding require many, many generations to see any kind of differences happen. And those differences don't just grow on an organism, it's a result of a mutation or just a genetic variety. And the last thing, and probably the most important misconception that students have, is they have a really difficult time reading dichotomous keys, not because they don't understand the concept of how to read it, but because they don't understand specific words within the dichotomous key. For instance, they might not know what webbed feet is, or a dorsal fin, or a complex leaf versus a simple leaf, or a membranous wing, or something, or anything like that. So specific parts of animals, sometimes students get confused. A fork in the tail. I thought that was pretty self-explanatory, but students had no idea what a fork in the tail was until I showed them what it was. So any opportunity you have to talk about specific animal traits and the weird words that might go along with that, like dorsal fin, it's important to bring it up because students might not necessarily have the vocabulary or background knowledge. Okay, project explanation. So the project that I've planned for this unit is very similar to how I started this video. It involves students assuming that NASA has found a couple of new planets, and in order to diversify the planet, they have to add some organisms. So the genetic engineers, in this case the students, would design and model an organism that would best fit that environment. So there are three planets to choose from. So students have a little bit element of choice here. They can choose one planet, and if you want to have students do more than one, if your students can write more or you have multiple days to do this, I suggest doing more than one, or more than one organisms per planet. And then what they do with it is, in this nice pretty box on the right, they can draw their picture and label parts of the organism that describe its adaptations for the environment. And then I have students write a paper describing why that organism is best fit for the environment. This aligns really nicely to the structure of essays that students need for 7th grade English and writing STAR anyway. So it's important at some point to see or to make connections across disciplines that in science you also have to write papers too, and the format you do that is very similar to how you write an expository essay. Moving on, there are three main experiments or activities that I wanted to talk about as far as shopping list goes. The first one is the Beak Lab. This one is very important. It's one we've done in the past. It's where students pick at seeds with popsicle sticks, acting as the big and small beak of a bird, and this helps them decide how natural selection, or see how natural selection happens over many generations. For the Beak Lab, you'll need popsicle sticks, seeds of some kind, paper plates, and probably a broom because those seeds go flying everywhere. There is what I call a bunny simulation. That's where I describe selective breeding. I have all of the students act as bunnies, and I'm the bunny farmer, and I kill off some bunnies and I multiply other bunnies to show them how I can increase or decrease a population of a specific trait or gene. For that bunny simulation, you don't necessarily need anything, but I usually make little bunny faces for students to hold over their nose so that they know what kind of bunny they are and whether they've been killed or not killed. And then the last thing is strawberry DNA. There's many resources, good resources online for how to do this. It is very important that you use cold isopropyl alcohol and not anything else, otherwise it will not work. I have had bad experiences before, but the students really loved strawberry DNA, and I have them make a necklace to put that DNA in at the end of the lab so they can go home with something really tangible. I can provide more resources for strawberry DNA and exactly what is on that shopping list in the unit plan if you check underneath resources and materials. The last thing I want to talk about was the end goals of Unit 3. By the end of Unit 3, your students need to be able to do the following. First, they need to be able to write a lab report including data analysis about natural selection, specifically beaks, because that's what the CA number 3 is about. They need to have a general understanding of the location and relative size of a nucleus, DNA, traits, genes, chromosomes. And then lastly, they need to be able to read a dichotomous key and be able to understand all the words inside of a dichotomous key. If your students can do that, they'll be very successful. If you have any questions or further questions about this unit, this should also say Unit 4, I apologize for the typo, please email me at alawrence@uplifteducation.org. Thank you.